Thanks, Afia. Okay, Rachel. Join us. Okay, so this week we're gonna learn about transcriptions and translation. Okay, how we transfer, how we uncodes how do we translate all this coding of DNA into a working protein form? Okay. So at the end of the topic, you all should be able to understand and describe the process of transcription and translation, the importance of specific protein synthesis, why it has to be specific and accurate. Okay, you all relate the function of protein synthesis with food. Okay, so what exactly is transcription and translation? Transcription is you generate an mRNA from a DNA. So basically, it's a type of copy from your blueprints. Okay. Translation is whereby you translate lah. Whereby you, how to say, macam you put into, from drawings you put into physical form, physical working form. Okay, that's how you do it. Okay, we are differentiate between these two things. All right, prokaryotes, simpler organisms, simultaneous transcription and translation process. Eukaryote, however, transcription and translation are different. It's not simultaneous, bukan sekaligus, whereby transcription takes place first. After it's finished, then translation takes place, because it is separated between space and time. So, and it requires more extensive processing of primary RNA transcript. So why is it these two between the bacteria or prokaryotes are different from eukaryotes, other than space and time? Okay, so this one, bacteria ni sekaligus, the moment you buat dia punya mRNA, terus buat dia punya protein. However, for eukaryotic, it takes time. You notice the structures? Why is it different? Guys, there's a question mark, yeah? I mean, I ask a question. Come on. Kenapa berbeza?
Maybe because the eukaryote is more complex.
Eukaryote more complex, exactly. So why is it is much, it's different. So what does it lead to? So more complex, so?
More organized.
So what?
So the process become more organized.
Process become more organized. Okay, due to the complexity, the process takes more time. Okay, and as the figure shows, for your processing of protein, your mRNA, there's a space, ya, ruang yang berbeza. Okay, space whereby this mRNA needs to be transported first outside the nucleus cells, right? Once, because your ribosome, all the component cannot enter the nuclear envelope. Okay, it's tightly lipped. But your mRNA needs to get out first. Then all these floating ribosome can attach to mRNA and then transcribe or translate into a working functional protein. Okay, this is why when it comes to bacteria, everything is just mixed up with even their punya DNA is just floating around. Okay, there's no specific space or ruang for your DNA. And as well, if you look at between bacteria and eukaryotic cells, what do you see in terms of its growth rate?
The growth rate between those two?
Yeah, between those two.
I think the eukaryotic cell is a bit slower.
Correct. Bacteria are much more faster. If you were to culture, put it on plates like a petri dish, bacteria can grow within a day. 24 hours you nampak dah ada growing on your media plates. You have seen media plates. But for cell culture, if you study if you know cell culture, or if you do macam COVID study tu, they require much more longer time because the cells are complex. Okay, it requires a lot of different things. Okay, it needs to be processed first, it needs to go out, it needs to do this, that, and that. Nanti we'll see why is it eukaryotic are much more complex. Bacteria is like more straightforward. So everything that happens within the bacteria are much more rapid. Okay, the growth, because the growth is much more rapid. The life cycle is much more rapid. Okay. All right. So in terms of RNA compared to DNA, RNA is more abundant. Of course lah, because although DNA are much longer, but DNA ini is just a master, is exactly a master copy. Okay, it's a master drawings, right? The RNA is copy from the master drawing. DNA is found in the nucleus within small amounts in mitochondria and chloroplast. RNA ini bersepah-sepah throughout the cell. Okay, and bear in mind, we don't have only mRNA, we have tRNA and mRNA. Okay, several important differences between RNA and DNA. Pentose dia, remember, RNA is ribose, DNA is deoxy. Okay, minus one oxygen molecule. RNA uracil, in DNA thymine. RNA is single-stranded, DNA is double-stranded. Okay, RNA molecules are much smaller than DNA molecules. Any other example? Any other example, the differences between RNA and DNA? Hello?
Someone wrote in there.
Oh, someone wrote it. Sorry. If you wrote it, I'm not sure whether I can check lah. Since I'm presenting.
Do you want me to read it, sir?
Yeah, sure. Why you wrote it? Just speak in the class lah.
I don't know. It's from Mufliha, and she said that DNA have helix shape while RNA doesn't.
Okay, in terms of shape. All right, good. What else? Oh, I can do those. Okay. Lagi?
DNA is stable in alkaline, but RNA is not stable.
Stable in alkaline. Oh, okay. That's new to me. Maybe because of the interactions. All right. What else? Lagi?
Doctor.
UV?
I searched the internet and it said that RNA is much more invulnerable to UV light.
Invulnerable to UV light, maybe. If compared to DNA, because DNA are much bigger, so the chance, like if you have bigger surface area, you much more exposed towards the changes. Whereby RNA is just short repeated of DNA of nucleotide sequence. Yeah, makes sense. Smaller means shorter. What else? Lagi?
Maybe the way it replicates, like DNA replicates, but RNA needs to be synthesized.
Sorry, how? What?
I like the way it like makes copy of itself. Like DNA can self-replicate, but RNA needs DNA to.
Yeah, it needs a references. Okay, it cannot replicate on, so it needs references. Okay, all right, that's good. Okay, so moving on. So we have three types, main ones. We have rRNA, mRNA, and tRNA. rRNA ribosomal is the major component part of ribosomes. Among of all the RNA exists within a cell, rRNA is contributes to the biggest percentage. Because why? RNA is the bigger size of all these things. Nanti nampak how rRNA works. Messenger RNA is the one that codes from DNA to RNA. Okay, the ones that carry the codings lah. Whereby the tRNA, okay, the second biggest one, mRNA is, although our mRNA paling banyak bersepah being coded, but because it's smaller sizes, so it doesn't contribute as much as the tRNA and RNA. tRNA ni dia bawa, those are the one that carry amino acids. Okay, all these is not a structural form of enzyme or proteins. It's actually a component of nucleotide sequence. Okay, like this, nampak ACC. Tepi-tepi ni, everything is nucleotide sequence. They are looping and looping lah, macam it gives a certain form base interaction of sequences. So they become macam a carrier. But it's not a protein, yeah. So don't get mistaken. Protein enzyme is not the all three of these. Okay, so mRNA carries a genetic code to the ribosomes from DNA. Complement codings, yeah. I explained to you. A codon is a triplet of bases along the mRNA that codes for particular amino acid. Like for example, this one lah, GCA, ACC, TAA, all that carries a certain specific amino acids. Okay, and it's come with a triplet. Tiga macam ni. One codon, one amino acids. Each of the 20 amino acids needed to build a protein has at least one codon. For example, that particular codon, that particular amino acid may have 1, 2, minimum 1, of course, can ada minimum 1 kan, 1 at least, and it can have up until 3 to 4. Okay, so we have 20 amino acids, and then they also start codon and end codon. So basically, if you later we kita tengok how much the combinations. So basically it encodes for 20 amino acid plus end codons. Okay, so start codon is actually encode for amino acids named methionine. So among the 20 amino acid, how many amino acid human can produce? Berapa? How many amino acid we can produce? What's the chat? Okay.
10.
Huh? How many acid we
is 12 grams.
Huh? Grams.
Kalau
Types. We have 20 types, kan?
hmm,
How many amino acid you can produce?
10.
10?
No. Okay. Do you remember essential and non-essential amino acids?
Isn't it 9?
9 is essential, which means 11 is non-essential. What does it mean by non-essential? Tak penting. Meaning that non-essential is the one that we can produce. 11. So we can produce 11 amino acid. The rest, where do we get the remaining 9?
Food.
From our food.
Yes. We acquire them from our food through consumption lah. Okay. All right. So there are also codons that signal start and end. Okay, this one. Okay, let's look at the combination. Okay. Remember that how many codons are how many nucleotide sequence that we have? 4 types, right? A, C, T, G. Codon is always in triplicates, right? So of all these, without counting, how many combinations that we have? 4 nucleotide sequence, 3 base. So berapa? Part kuasa. Part kuasa? 1. 3 lah.
Yeah.
So how many combinations in total?
64?
All together? Huh? 64,
64.
right? So we have 64 combinations that encodes for 20 amino acid. But wait, remember that we have start codon and stop codon. Start codon is fine because start codon start ATG, only one codon ATG, which is stand for which encode for methionine. However, we have 3 types of stop codon: TAA, TAG, and TGA. So basically, only 61 combination that encodes for 20 amino acids. Okay, and only these two, methionine and tryptophan, only encodes for 1 codons. Okay, so these two are very specific, especially for your start codon, methionine. Methionine always has to be ATG. Sorry, ATG always has to be start. any kind of sequence of protein always start with ATG. Okay, always. That's what we discovered lah. Okay, so this is how it goes. Nanti I'll show you. I'll share with the video, or just let's watch a video je lah. Nanti I'll explain it again. Where's my Safari? All right, can you see my Safari? Yeah. Okay, this is the most simpler one lah. Just watch it very quickly, yeah.
DNA strand contained.
Can you hear the voice?
Are contained.
Dengar tak voice dia?
Dengar tu.
Okay, okay, go.
In our DNA, DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with the promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as the 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strain that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins.
Okay. Okay. I think that's pretty clear, but I need to explain a little bit more detail.
I'm a scientist and I do
Go
not
on.
have a lab at my disposal. I hope
My
that
YouTube's still talking.
everyone-
Hold on, yeah. All right. Okay. So if you look here, yes.
Ada orang dekat waiting room nak
Ada
masuk.
orang kat waiting room pula. Okay, Afara. All right. Done. Thanks for notifying me. Ini kalau duduk luar kelas ni mesti tak masuk dah kelas dah ni. Okay, here we are. This one. So DNA double helix unwind near the gene that is getting transcribed. So basically, when they start to transcribe a certain region, okay, this DNA start to relax. All right, but because as you understand, DNA is a very, very coiling and compact sort of structures. So once a certain genes needs to be transcribed, it start to macam terurai, right? Unwind to become much more relaxed so that your RNA polymerase can bind to a certain places. Okay, as you noticed it before, all right. When it comes to transcribe certain genes, it has a promoter and it has terminator. Sama jugalah, it's like one is the entrance, one is the exit. What's important having to have promoter and terminator? So these two regions are recognition site. Okay, macam RNA polymerase is sort of macam blind. You cannot, okay, macam nak tahu where exactly the gene to kat mana from where to where. Basically, it doesn't know. All they being coded is for RNA polymerase ni, dia nampak sort of macam, okay, dia nampak DNA to start to unwind. Once unwind, dia, oh, boleh masuk ni, macam kedai dah buka, right? So kedai buka, but which shop we want to go? Because that particular DNA, when it's unwind, there's a lot of genes in there. But where to go? So particularly, it recognizes certain places whereby macam kalau masuk kedai, you know, ada promoter tu, masuklah, tengoklah barang saya. Okay, sort of like that. You go to a certain entrance where your RNA polymerase only recognize that particular promoter to dock. Once you recognize, then baru dia buat kerja dia. Dia transcribe one by one nucleotide sequence up until terminator. Okay, nanti kita explain. Nanti I'll share a bit detail on how they exactly start and stop. Okay, it's a bit more complex than that. So it says the transcript use 1 or 2 exposed DNA strand as the template. This strand is called template strand. Okay, whereby the template strand is a complementary to the template strand, identical to the DNA strand or the coding strand. Okay, non-template or coding strand. So let me just explain it quickly. Hold on, I think I can use some pen. Oh yeah, annotate. Draw. Okay. Here. So here, okay, template strand. Yeah, this one, whereby your RNA binds, but truthfully, your RNA will be identical to the coding strand. This one. So this one yang dia copy, because since it tak boleh copy directly, it is complementary. So the RNA polymerase bind to this one, so that whatever the copy that they make is exactly the same as this one, but except for T and U lah. G sama, A sama, blah 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 blah. Okay, so your everything is just complementary. This is called coding strand or non-template strand. Okay, and finally, and then this one is transcribed from 5 prime. This one is 5, yeah, 5. Oh, so ugly. 5 prime, 3 prime. This one is 3 prime, 5 prime. Okay, so your RNA is exactly identical to your coding strand. Okay, so requires DNA template, nucleotide, and magnesium, and certain enzyme factors. Okay, that one after that I explain in detail. So clear, yeah? So you have to be clear when you talk about template strand, coding strand, or non-template strand. Okay, it's always been, it always is a complementary. All right. Okay, what is it there? How to delete, delete clear all drawings. Okay, good. Okay, this one. Here we go. So it's like 3 prime to 5 prime. Your template, your RNA is growing from 5 prime to 3 prime. When anytime you want to make anything from either from DNA or RNA, the construction always 3 primes are the one being added all the time. Dia tak boleh reverse, cannot be reversed. Yeah, all this addition nucleotide sequence comes from the 3 prime. So this is what called initiation, okay, especially in bacteria. RNA polymerase binds to DNA at a gene origin called a promoter. Promoter ni bukanlah tulis gitu, oh ada promoter. No, exactly. A promoter is basically a sequence, like a type of nucleotide sequence that encodes for something. Usually it's called a tata box lah, or certain nucleotide sequence. Okay. Promoter DNA sequence that let RNA polymerase or its helper protein to attach to DNA. Because basically, you punya start of coding, what you punya start of codons, you still remember your start codon apa? Ingat tak? The triplets tu. ATG. Okay. Your start codon ni, your RNA polymerase start kat sini terus. No, it won't. It will start further up before that. Okay. So in bacteria or prokaryotes, RNA polymerase recognize and binds directly to negative and 35. So negative 10 and negative 35. Here and here, DNA sequence elements. It's called upstream regions. Okay. This is your coding. This is called upstream. Whatever down here is downstream. Okay, the binding to our DNA requires a sigma factor that falls off after 10 is found. So basically, your RNA polymerase ni, dia jumpa this sequence. They attach at the same time your sigma factor help to attach. Usually sigma factor will bind first, and then once your sigma factor binds, and then you punya RNA polymerase nampak, there's a sigma factor there. Kawan lah, dia dapat kawan, jom lepak sekali. So they lepak together here. Once it attached together here, it form a complex. So dia baca, dia gerak, dia just sliders ya. It slides. Let me just hold it. So it slides. It found here. It slides. Dia gerak lah. Cucu train gerak all the way here. Once all the way here, you recognize tata box or -10. Then your sigma factor starts to. This is sigma factor. Start to falls off. That leaves you only RNA polymerase, and then it start read baca semua from here up until everything here. They start to encode and start to produce elongated mRNA. Your mRNA bukan start here, okay? The moment dia jumpa sini, dia dah start dah. Especially the -10, it will start to add nucleotide bases. Okay, your mRNA always doesn't start. Hold on, where's my mouse? Am I lagging?
No, doctor. I'll tell you if you're lagging.
Oh, okay. I'm not lagging, but somehow my computer is lagging. Oh, where are you? I can't find my mouse. Sorry. Where you are? Where are you? Sorry, technical error. Okay, stop share. All right, there you go. Let me start share again. Okay, so a bit clear, sigma. All right. So in eukaryotes, in humans, eukaryotes, the -10 is called tata box, almost similar as your prokaryotes. Okay, but no sigma factors, but require transcription factors. Okay, when it comes to transcription factors, if I were to explain in detail, eukaryotes punya transcription is very, very complex. It requires a lot of bindings, okay, of transcription pattern of protein factors. This one lah, yang nampak macam ada kecil-kecil ni kan. This is the most, I could find the most simplest form that can be used to explain in very briefly. Okay, so this one, instead of sigma factor in prokaryotes, your eukaryotes has a lot of more friends that are required so that your RNA polymerase can be bind. The reason why tata box is one of these, I'll call it promoter region, promoter site. Tata box ni a bit, the binding of tata box will create a binding of AU. Okay, remember that if AU or AT, dia punya binding is not as strong as compared to GC. GC has 3 bonds. AU ni, TA or AT or AU ni, dia punya bond is 2. So not as strong. It's sort of like weak bonds lah. Okay. So when it comes to eukaryotes, okay, your transcription factors recognize this site, start to bundle up a lot of transcription factors, and then together it binds with RNA. Recognize, ada transcription factors. Ini macam nak transcribe je ni. So they will follow, bind all together, and then the transcription factor stays, and then start to read whatever the sequence here up until it recognize the termination factors. So my question would be, what is negative value and what is positive value? Okay, when it comes to coding, your start is ATG. Always, remember, always start with ATG. So the moment you found ATG, A is your 0 point. T is +1, G is +2, meaning that you are moving towards this right side. Okay, anything you move from 5' to 3', that's called moving towards the positive side. Whenever you move on from start of A, move on to the left side towards 5', that is called minus sign. Or in other words, anything that is minus A or before the A region here, hopefully it doesn't lag. Okay, A, T, G. Okay, whatever move here, negative. Whatever move here, positive, right? So this region of negative also known as upstream. Okay, this region here, whatever T starts from T is 1, whatever that comes here towards 3' is positive. So when I say 5' and 3', always related to coding sequence or coding nucleotide, not the template. Yeah, the template will be opposite this one, 5' to 3'. Okay, so don't get confused. Clear? Any questions before I move on?
Hey, doctor, the template is 5 to 3', right?
Oh yeah, no, the template is 3' to 5'.
Okay.
The non-template is 5' to 3'. Non-template is all equal to coding, because your RNA is exactly same sequence as your coding. But RNA polymerase doesn't bind with coding; it binds with RNA with your template or non-coding sequence. Faham tak? So don't get confused, ya. Alah, jadi lagi. Clear. I cannot draw some. Okay, clear? Any questions? Okay, this is where elongation. Okay, elongation RNA strand gets longer. Of course, it gets longer so that the addition of nucleotide sequence. So RNA polymerase walks to it baca lah. They baca all the sequence like you saw in the video along template strand of DNA in the 3', 5' direction here. They baca sini. Whereby it needs, because it needs to create similar sequence, exact sequence to your 5'. Okay, sama. All right. So topoisomerase in your eukaryotes, okay, higher organisms, its function is to relax or to unwind this coding sequence or this DNA double helix, because your DNA double helix is very compact and it's very at the same time it needs to be unwind. If it doesn't unwind, so how on earth your RNA polymerase can add sequence at the same time read the sequence? So it needs a topoisomerase. Topoisomerase usually kat sini. Dia macam driller. Dia cucuk buka, unzip here, at the same time it zip here. Okay. In system of transcription, it won't, your DNA tu dia takkan unwind semua and terbuka semua. It just does not it doesn't make sense, because your DNA, although it needs to unwind and relax on one point so that gene can be read, it cannot unwind everything, because if it unwind everything, it become much more loose, then it gonna sort of take a lot of space. So that one is not pretty rationalized. So it needs only to unwind a specific place whereby at that time your gene is being transcribed. Okay, so in terms of in bacteria, this is not called as topoisomerase, it is called gyrases. Gyrases sama rule of gyrator, macam driller. So it drills here so they can unzip. So the back follow will zip at the same time. So bergerak lah. Okay. And your mRNA will keluar like this. Sort of lah. Okay, in termination. So RNA polymerase will transcribe until it reaches termination site. Transcription stop point. This is for eukaryotes. Actually, eukaryote, if you look into online or you can YouTube it, Google it, it's not as easy as sampai situ je, oh stop, habis. Macam dalam video tadi, oh nampak RNA termination site, termination stop point. It stopped and then it's just released. Whereby in much more detail, a lot of players, a lot of factors, the bindings, the factors, at the same time, the combination of this one, the interaction between RNA polymerase and transition stop point sort of get weaker. So it forms a certain complex. Okay, you can Google it, you can start a certain complex, so that at the end of the day, RNA polymerase will have weak binding with your DNA, and then it just finally dissociates on its own once it reaches the stop point. That's the most simpler form that I can explain. However, in the bacteria, there are two different types. The first one where I want to say row-dependent terminations or row factors, okay, RHO. Row factors ni, at one point, your DNA, your RNA is being transcribed, okay, it's grow. It has a certain binding or sequence that row can recognize. I can't remember. It's like TGG, TA, not like Tata, I'm not remember. They have its own sequence. Row factors ni, they recognize. Yeah. Once it recognize, they can follow. They can like climb together. Your RNA polymerase will move towards here. At the same time, row ni dia panjat up to a certain point. It will catch up with your RNA polymerase once it reaches stop codon. If it's not reached stop codon, it will slowly catch up. And then once both of these RNA polymerase sampai kat termination site or stop point, your row factor will catch up. Once row factors catch up with RNA polymerase, it becomes a hybrid complex factor. At that point, it starts to become weak, because at the same time, here as well ada macam a tata box sort of like AAAAU, ada macam like AAAT sequence sort of like that. This AAU sequence pun juga is a weak binding. Together it form a complex. Somehow it weakens the bind, and finally your RNA polymerase sort of macam falls off from the transcription. So it stops lah. So in terms of transcriptions, it has to has a stop point. My question would be, kenapa it has to has a stop point? And after, kenapa dia tak transcribe past the stop point? Biar lebih sikit. Kenapa ada stop point?
Nanti terlebih transcript.
So what kalau terlebih? If your mRNA, katakanlah your mRNA is like this, you have your start codon, which is ATG, and you have stop codon. Kalau lebih, doesn't really matter, because your translation later on, every time dia jumpa stop codon je, dia akan stop. It won't go past that. Right? So kalau lebih, tak apalah. Right? Once it has to have a stop point. Any idea? Stop point and you punya stop codon tak sama. Don't get mistaken. Your stop codon might be somewhere here. Here or here. This is called termination site. So your genes maybe from here to here saja. This is not stop codon, ya. So don't be mistaken. That's why I said, kenapa nak kena ada stop point? Kalau dia baca sampai habis, yalah. What's the problem?
Maybe so that the RNA polymerase can detach and do other DNA, maybe.
Yeah, for detached. Tapi kenapa it has to be stop codon, kena ada stop point? At one point, if this row factor will catch up, it was just dissociates, right? That's also true, what you said. One of the reasons being it has to have a stop point, because why you want to waste all your resources, like nucleotide, because every time you add here requires energy. Requires your nucleotide. All this nucleotide is your resources. Macam nak buat rumah, batu bata, right? It has to have a raw material in order for you to make this. Why you want to waste your material to make something that will not be translated into something functional? Waste of time, waste of resources, right? So it has to have a stop point so that it knows when to stop, and then all the other resources can be diverted into other genes that needed to be transcribed and translated. Okay. And there's another point. There's another sequences. There's another what I call that, ways on how bacteria has stop point. That is through the creation of hairpin. Tahu tak hairpin tu apa? It literally looks it's a looping of your mRNA. Okay, it looks ni because there's an interaction between, katakanlah if here ada G, right? Here C. What do you think happened between these two?
They bind together, don't?
They bind together. Once they bind together, it creates a loops. Okay. At one point, if the sequence is much more similar, it can create a hairpin. Okay. Find out. Okay. I'll ask next time. Let's find out how does hairpin able to stop a transcription process. Okay. Find out, but nanti I'll ask you guys randomly. So make sure you prepare. Yeah. All right. Post transcription. When it comes to bacteria, post transcription meaning it needs a certain process. Okay. For bacteria, like I said, because it's simultaneous, it does not have any post modification. Whatever sequence that's within the mRNA is being translated directly into functional protein or polypeptide or enzyme. However, in eukaryotes, it is known as pre-mRNA. Mana is immature, tak siap lagi. So it needs to be further processed so that it can be a fully functional mRNA or mature mRNA. So usually this is needed because the 5' receive a modified methylated guanine cap. Here, G. This is used for stability, transport, and ribosomal attachment. Stability and transport is very important because your mRNA needs to be transported out the nuclear envelope and then onto the cytoplasm. So since your exon are much complex, much larger, okay, macam you bayangkanlah you ada lidi pendek, lidi panjang. If there's no stabilizer, okay, lidi pendek is fine lah, pendek je. It's kind of like stand like this. But the moment your lidi becomes longer, what do you think happen? Wispy, doesn't it? Lembek. So it creates instability. So somehow they create a G cap so that your mRNA are stable, and then once transported outside the nuclear envelope in the cytoplasm, it doesn't dissociate or pecah. So it's being guided, is being protected and guided by guanine cap. Right? At the same time, this area of region is being recognized by rRNA or ribosomal attachment. For a 3', sama juga, they call poly-A tail. Poly means multiple, plural. A is the adenine. Okay, instead of G, they put A. Sama juga to transport outside the nuclear envelope and for stability. So you need to remove introns using spliceosome, like I showed you just now in the video, and pasting together coded sequence. Okay, for introns is non-coded, exon is coded. So it needs to get rid of all these, so that it combine. All these are coded sequence. Macam ni ada coded, non-coded, coded, non-coded, coded. Okay. If intron doesn't splice out, what do you think happened?
Unstable.
If your introns does not being spliced out or cut out from the mRNA, what do you think happened?
Doesn't it make the mRNA too long?
It's fine, it's too long. Long is not the issue. If that particular protein or enzyme, it has very complex arrangement of amino acids, it doesn't really matter. Length is not the issue.
Maybe the sequence wouldn't be like correct, I guess, because it's not together.
Exactly. So now, supposing your sequence, because in terms of protein and enzyme, like I said, because of its interaction, any edit sequence that is unnecessary or is unwanted could disrupt the structure of the finished product. Sama macam your class lah. Okay, your class is already good as it is. You sit where you are, you sit where you like, with your friends or with your away the ones from you hate, close to one you love, or from boyfriend girlfriend, you love each other. The moment ada certain people masuk within your class, it's gonna change the whole dynamic of the class. Tiba-tiba masuk ni macam kecoh gila, or like very loud kind of person, or dia ni jenis yang rampas girlfriend orang or boyfriend orang. It disrupts the whole dynamic, doesn't it? It changes everything. If it changes everything, and it becomes a different kind of proteins or non-functional proteins. Okay, so in terms of this, introns needed to be accessed out or cut out. Okay, become functional. Okay, RNA capping, sama je guanine or phosphate capping. Usually add metaguanisine shortly after RNA polymerase and polyadenylation. Okay lah, guanine cap or 5' cap or poly-A tail cap. And the coding sequence, it needs to be cut. Got one single unit of coding sequence, okay, and protein. Like I said macam tadi lah, okay, in terms of when you transcribe your DNA to mRNA, so here is the promoter start binding. Here is the start of your coding sequence. Here is the stop of your coding sequence. Finally, here, this is your terminated region. Like I said, your terminated region with the DNA does not mean that this is a stop codon. Stop codon and start codon is actually inwards, okay, of your whole mRNA. Dalam lagi. Faham ya? Faham tak? Okay, in bacteria, the RNA is translated into protein. Okay, in eukaryotic cell, the primary transcript is made by coding sequence exon and introns. Have you ever wondered where introns go, or why exactly this exists of introns? Only exons are the one, that is actually being translated into functional protein. So kenapa ada introns? For what introns ni? Untuk apa? Any idea? When it cut out macam ni, bila dia cut kan, hanging macam ni. So untuk apa? For what? Why exist in the first place? Since it's very so much useless. Tak function pun. Any idea?
Maybe to allow a cell to choose which exon will go into the particular mRNA.
Sorry. Macam mana siapa ni? Who is this?
Yeah, Iliani.
Iliani, okay, Iliani. Right.
Maksudnya macam nak allow apa ni nak allow cells tu untuk pilih exon yang boleh bind together.
Alright. Kenapa our DNA, especially eukaryotic, kenapa tak macam bakteria aja? So if it's like bacteria, there's no intron and exons, right? Terus the code for everything that is coded is actually a working or functional protein or functional coding sequence. Kenapa ada non-coding sequence pula? Kenapa? Why it exists in the first place? Google, man. Alright, I'll ask next class, yeah? So you better Google it. So ada dua sort of things that you need to Google. So find out what introns are for. It's actually very, very interesting. Yeah. So okay, just give you one hint. Introns go to here. You notice this snRNA. It's very small and it creates sort of like a certain shape. Macam this one like sort of like happy loops lah. Okay, these happy loops, okay, unwanted codings, which is introns, okay, may or contribute into small nuclear ribonucleotide protein, or called that SNPs or snRNP. Okay, so they are the one that is exactly involved at the same time involved. Okay, you can see here. Oh, apa tu nak tampak? Hold on. Screen share. Okay. So this one, SNPs, snRNPs. Okay, it creates certain loops. All these are RNA sequence, coding sequence. So it has the ability to bind to a certain area of your DNA sequence, and this can attach and bring a loop so that your spliceosome, which is actually an enzyme or protein, can cut your intron, and your intron will be recycled to be used, not wasted, yeah, cycled to be used. One of it is called snRNPs. So you can Google snRNPs, the function of snRNPs. It's very, very interesting. And scientists nowadays have discovered a lot of the use of snRNP as much important as your own DNA. Okay, snRNP. Clear? All right. So why you need to splice? That's another reason why you need to have you have to have your introns. Okay. So this is called a lot for genetic recombination. Recombination you can recombine a different patterns, right? A different patterns of your mRNA that can create a new things. Okay, it says here also allow one primary transcript like mRNA to encode for multiple proteins by rearrangement of the exons. So basically, if you rearrange exons in a different kind of way, once been transcripted and you have exons and introns, all these create a different type of variety of muscle. One gene, one coded gene, your intron and exon can be processed in a different way. It can create different kind of functional protein. For example, this one, it can create striated muscle cell or muscle mRNA. This one can create smooth muscle. This one can create fibroblast, okay, one component in your bones. And this one is called for brain cells. Sorry, not brain cells. Brain muscle cells, sort of like that. Okay. So it creates a different kind of variety based on the processing of introns. It's like, if I were to compare, it's like having a piece of paper. You want to do origami. That one piece of paper, you can fold it into different kinds, make it into a swan, make it into a box, make it into an aeroplane, and blah, blah, blah. But the base is all same, but it's just how you process into different kind of things. Right? Okay. Okay, let's hold on. Oh, okay. Can we take 5 minutes here? All right. When we come the next 5 minutes, can I have 2 volunteers to share your fun facts? Can
Can, doctor.
And
two people share your fun facts, and then we'll join again in about 5 to 10 minutes. Is it okay? So you can
Okay,
take a breather
yeah.
a bit, then we continue. All right, see you all in a bit.